Hello everyone, my name is David Silver. I'm a professor at Duke NUS Medical School in Singapore, where I also serve as the Deputy Director for the program in cardiovascular metabolic disorders. My laboratory has a long-standing interest in understanding the function of lipids in human brain health. And what I'd like to do in this brief presentation is tell you about some of our findings that we've generated over the past decade, in which we've identified a very key lipid that's important for maintaining brain function and health. Aging is inevitable, but what I'd like to tell you is that there are systems in the body that prevents cognitive decline. Normal aging, as we know, it generates reactive oxygen species. These are major drivers for brain aging. And so in this cartoon, I'm depicting healthy neurons on the left side, and during aging, some of these neurons get damaged just through the process of normal metabolism, which, as I mentioned, generates reactive oxygen species that damages these cells. However, the body has mechanisms to bring this process back into homeostasis and prevent excessive cell death of neurons. Now, one of the systems that the body has to revitalize damaged membranes in the aging human brain is to replace membranes that are damaged. And the brain does this by utilizing a very important lipid called lysophosphatidylcholine omega-3, or LPC omega-3 for short. This is the chemical structure of an LPC omega-3. So just briefly going through the structure, we find choline, this is the C in LPC, followed by phosphate, glycerol, and the omega-3 fatty acid DHA. These LPCs are critical precursors of membrane building blocks in the brain. And one of the key discoveries from my lab is that LPC DHA is the primary molecule that the brain uses to obtain omega-3 fatty acids, and it does that by transporting in at the blood-brain barrier LPC DHA via a transporter called MFSD2A. And my lab solved a high-resolution cryo-electron microscopy structure of MFSD2A protein binding to an LPC DHA, shown here in this magenta structure in the middle of the protein spinning on the screen before you. So this is essentially the door that allows LPC DHA to cross the blood-brain barrier and enter into the brain. And these LPCs are the primary way, or the form, the chemical form by which the brain obtains omega-3 fatty acids and membrane building blocks to replace damaged membranes. Now, LPC DHA is a natural product. It's made by the liver. So if you are eating plant material that contains omega-3 precursors, such as linolenic acid, the liver can synthesize DHA from linolenic acid. Then it packages that DHA into an LPC molecule, which leaves the liver and circulates in blood. It reaches the blood-brain barrier where this protein I just mentioned called MFSD2A lives. And this is the protein that brings LPC DHA across the blood-brain barrier into the brain where the neurons are. Now, I'd like to tell you some distinctions between LPC DHA and some of the known dietary supplements that contain DHA that you can buy in the marketplace. And I want to tell you about their metabolic fates to convince you that LPC DHA is the most bioavailable form of DHA, in addition to the fact that it serves as a precursor for membrane building blocks to replace damaged membranes. So in this diagram, I'm showing on the left side some of the common types of dietary DHA supplements that you can purchase in the marketplace, such as fish oils, which contain triglycerides containing DHA, and phosphatidylcholine DHA, which is essentially krill oil. So if you ingest either of these two forms of DHA, triglycerides or phosphatidylcholine, they are digested in your gut by pancreatic lipases to liberate DHA. This DHA gets repackaged into triglyceride in your gut. And that then gets packaged onto lipoproteins and enters into the bloodstream. where some of this DHA gets dumped to adipose tissue, heart, skeletal muscle, and a small amount comes back to the liver, where the liver can then repackage that into LPC DHA, where it circulates in blood on albumin. And this LPC DHA gets transported across the blood-brain barrier by the protein I mentioned to you called MFSD2A. Now you can imagine if you take a dietary supplement containing LPC DHA, that this would provide a direct route to the brain, bypassing this circuitous route of delivering DHA to various organs. Instead, LPC DHA would be delivered directly to the brain. So this is the kind of system that the body uses to obtain DHA, as well as replace damaged membranes in the brain. So you can see that regardless of the type of dietary supplement that's available that you would take, triglyceride, DHA, or phosphatidylcholine, they all tend to have the same metabolic fate. The unique feature of LPC DHA is that it's directly transported into the brain. What is the evidence then that LPC DHA is the most bioavailable for brain uptake? And that's shown here in a paper that my laboratory published in 2014, where we had animals that were given DHA. This would be kind of similar to what you get in the marketplace for a triglyceride oil. This is called unesterified DHA. It's essentially just the fatty acid, versus animals that were given LPC DHA. And on the y-axis here is the amount that gets taken up by the brain. And you can see that LPC DHA gets taken up by the brain, whereas the unesterified DHA largely does not. And so this is very good evidence that LPC DHA is much, much more bioavailable than DHA. And there's other laboratories, in addition to mine, that have very similar evidence that has been published that LPC DHA is the most bioavailable form of DHA for the brain. Now, is LPC DHA important for brain development and function? And my lab over many years has identified families that have mutations in the transporter. So they do not have the ability to transport LPC DHA into the brain. And what we find in all of these patients, 100% of these patients have underdeveloped brains, or what's called microcephaly. And 100% of them also have developmental delay and intellectual delay. This underscores the importance of LPC DHA in brain function and development. And this provides probably the strongest evidence that LPC DHA is extremely important for the brain. And no such evidence exists for other omega-3 fatty acid dietary supplements. Now, is this important during aging? We know from work out of Stanford, this is a seminal paper published in 2020, in which this group at Stanford studied the aging blood-brain barrier. And one of their major findings was that in aging, the level of this critical transporter for LPC DHA at the blood-brain barrier is reduced. This implies that the natural uptake of LPC DHA to the brain becomes limited over time. This provides a very strong case for dietary supplementation using LPC DHA during aging. So if the transporter that transports the lipid into the brain declines, there's an opportunity to increase the amount of LPC DHA that the person would take, kind of flood the remaining transporters that are at the blood-brain barrier. So this is a very good argument for dietary supplementation. So I'll leave you with this last slide, which has really the take-home messages for you on the potential benefits of LPC omega-3 supplementation for brain aging. One is that LPCs, being precursors for membrane building blocks, are important for replacing damaged membranes in neurons during aging. LPC is also the most bioavailable source of omega-3 fatty acids for the brain. And it is also the physiological source for choline for neuron function. I didn't have time to go into a lot of details here, but this is the major source that the brain utilizes for choline uptake. So thank you for listening. And many of our papers are publicly available. So I would encourage you, if you're interested, to delve deeper into the biology and biochemistry of LPC, DHA, and MFSE2A, to go and read our papers. Thank you very much.